Good day, my dear postgraduate students. Welcome to our next class on histology for pathology. And in this class, we shall be dealing with pancreas. And please do remember, it is for budding pathologists. You people should be as familiar with the histology so that you pick up speed. So this is a sketch. I'm finding the duodenum, the different parts of the duodenum. We will not go into the anatomy proper. So there is a pancreas with a head, neck, body, and tail, and there is an aorta in the inferior vena cava. And what is draining into the various blood vessels? You people can see over here. The same thing is shown. So there is a stomach. In fact, there is a dotted line because these are all structures which form the stomach bed. That is what my professor of anatomy, Dr. Cooper, used to describe as. So this is the pancreas. It has got a lobulated appearance that I can make out. And there is a free hanging tail, which will be actually making an impression in the spleen. Have an idea about this. Now comes the important one. So I find that it is a lobulated structure and it is somewhat, I won't say pyramidal, but leaf-like in outline. I'll go to the anatomy at a later date. The gallbladder, etc. And along with the pancreatic duct, they drain into the ampulla of Vater over here. That is shown. What I'm interested in, these are all the pancreatic glands over here, and this is a duct. And this is a supporting stroma. And acinus is one. So look at this one, it is a gland. I hope you people are able to make out one gland or one acinus. It is lined by somewhat pyramidal cells. The nucleus is basal, and there can be some centroacinar cells also. And this is the lumen of the gland. They will be draining into a duct tube, and the ductules can join together to form a larger duct. That is the basic histology you people should have in mind. I hope it is very clear. The acinus, the lumen, and the duct tube. See what is happening over here. At this magnification and sections, I will not be able to see all the things. They will be cut here and there. These are all the groups of pancreatic acini, and they are supported by loose connective tissue. This is slightly different, large, rounded, paler, and these are the groups of islet cells. And here I am able to make out some kind of a septum. Because always the septum are important, they will be carrying the blood vessels. Again, a beautiful diagram over here. There is the acinus, triangular. There are some granules at the apex, which are called the zymogen granules. And these are the centriacinar cells over here, intercalated disc or the duct, I'm sorry. That is one, because it arises from the acini as such. So the pancreas, very easily you people can make out, and definitely it will be when you see a group of these islet cells. Otherwise, it is closely packed acini, and I'm finding the basal nuclei. The apex is somewhat pinkish in color, and I'll be lucky if I can see a granular picture, the zymogen granules. Look at this over here. There is a granular picture over here. Those are the zymogen granules. The entire thing, one here is, see an imaginary line. It is an acinus, and then there is an intercalated disc, a duct. So look at this. So the intercalated duct is over here, leading to interlobular duct, and then the main duct, acinar cells. Intercalated duct is over here. It is lined by the ductular epithelium. There is the endoplasmic reticulum, etc. You can forget. Hale's histology, beautiful picture. Nucleus and then a granular cytoplasm because of the zymogen granules. And this itself is the islet cells. Islet cells, as I told you, are pale. It is an endocrine organ. An endocrine organ will be having a uniform appearance. It is called a somewhat uniform, organoid appearance. See, when you see one region, you need not see another. And the catch is there won't be any ductule or duct. It directly empties into the blood. The capillaries, for example, that is where all the secretions are being emptied. So this is the islet cell of Langerhans. And there are two types of cells over here. One that secretes the glucagon, and on the other one alpha that secretes the insulin. This is the immunohistochemistry for it to identify what kind of the cell that is proliferating. To Santak.